My name is Michelle McGlone. I am the director of the Brookline Adult and Community Education. And I'm so very excited and pleased to have actually a former student of mine, used to be a teacher in Brockton, and Dr. Shapiro was a former student of mine. And when the COVID virus broke out and I heard that it potentially originated with bats, I thought, well, I'm going to reach out to Julie. She's an expert in bats, and see if she'd be willing and able to join us. And she actually is joining us today from France. So it's late in the evening, and I really appreciate her taking the time to do this. Aside from her being just an amazing student, she went on, she was a valedictorian and went on to Harvard University and studied Romance languages and literature. And then she went on to get her PhD at the University of Florida, and she's an ecologist. And she studies disease, wildlife, and the effects of anthropogenic change using field studies, molecular techniques, and modeling. She is currently a postdoctoral researcher at the National Institute of Health and Medical Research in France. And I printed out her CV, and it's like 7 or 8 pages long with all of these wonderful publications that she's put out there, and just, like I said, has an amazing background. And I'm just so pleased to have her with us today, to share some of her knowledge, you know, about bats and public health. I'd like to go ahead and turn things over to Dr. Shapiro, who has, like I said, some great knowledge to share with us. So Julie.
All right, well, thank you so much. I was not expecting all of that introduction, so I'm blushing here a little bit. I wanted to just share screen to get my PowerPoint up so you guys can see some images, which makes it a lot more interesting than just listening to me go on. Let's see. Oh, share screen. Let's see. Okay. So you guys should be able to see that first image. All right, and now it's in the full screen mode.
Yep.
Cool. All right, well, I'm gonna just stand up here because I find it a little bit easier to present if I can stand and walk around a little bit, even though I'm doing this from my home here in Lyon, France. And I just really want to thank Mrs. McGlone. She's already mentioned she was my high school history teacher, but I'm just really excited to get the opportunity to talk to you all today. And I'm going to be talking about some of my different lines of research on bats and antibiotic resistance and how ecology can help us understand a changing world. And I want to thank you all for also being here today. So I'm going to start by talking about bats. They're one of those animals whose reputation perhaps precedes them. People might find them to be scary or maybe mysterious, but for me, they are really cute, and they can spunky and pack in a lot of attitude, like this southeastern Myotis. Now, bats are a really diverse group of animals. There's the second most diverse group of mammals in the world. There's over 1,400 different species, and we can find them on every continent except for Antarctica. And these bats come in a variety of shapes and sizes. They have different feeding habits. They live in different places. And it's also really important to remember that these bats can be very different one from another. So all bats belong to the order of Chiroptera, and that's similar to example to the order Carnivora. And so one species of bat might be as different from another species as your pet cat is from a grizzly bear. But because there's so much of this diversity, they're just a really fascinating group of animals to study. And these are all pictures that I've taken in the course of my studies and field work. And bats also provide a lot of really important ecosystem services to people. So they can disperse seeds, and that can help with reforestation and maintaining healthy forests. Some bats pollinate plants, including the agave that we use to make tequila. And bats also, a lot of bats are also insectivorous. They eat insects, and so that helps control insect populations, and they might even eat agricultural pests and insects that can carry human diseases. Now, I had my first up-close and personal encounters with bats now 15 years ago as an intern at Mammoth Cave National Park in Kentucky. And this was the first time I'd ever seen a bat up close, and I was really fascinated and smitten by them. The projects in the park were based on monitoring the different species that were in the park as well as their populations. Now you might notice in this picture of me here, those are not my hairy arms holding up the bat. That's actually one of the other scientists at the park. Because I didn't know that I would be working with bats beforehand, and I hadn't received a pre-exposure rabies vaccine. And so I wasn't handling or touching bats myself. But nevertheless, I was still able to help setting up the equipment, writing down the data from each bat as we took measurements, and organizing the samples. Now, the next year, I ended up working for the Bureau of Land Management in Oregon, and this time I did have my rabies vaccines, and so I could handle the bats. And again, the goal of this project was to understand the diversity and populations of bats across the state of Oregon. And this project actually continues to today and has expanded to Washington and Northern California. In 2012, after I finished my undergraduate degree, I went to Brazil as a Fulbright scholar. I was working at the university, the Federal University of Mato Grosso do Sul in the central west of Brazil. And again, here I was still focused mainly on the conservation of bats and understanding how human disturbance and urbanization can affect the diversity of bats in and around the city of Campo Grande. But my advisor asked if I might be interested in studying Leishmaniasis in bats. My supervisor, Marcel Bourguignon, studies bats, but his wife, Adriana, was a researcher of Leishmaniasis in humans, and there were also other researchers, including Manuel here, who I continued to collaborate with. Now, at first, I actually was not super keen on this project. I didn't know much about Leishmaniasis, and I had never worked with diseases in bats. But I'll give a bit of information about this disease. It's a neglected tropical disease. It has a worldwide distribution, as we can see in this map here. The darker the color, the more species of the parasite that causes the disease Leishmaniasis there are in the country. Now, this disease is can be caused by about 20 different species of Leishmania. That's a protozoan or a one-celled animal. And the disease is transmitted from individual to individual by sandflies. The sandflies, it's also a zoonotic disease, so it's naturally found in wild animals, particularly opossums and wild dog species. But in urban areas, domestic dogs are actually an important host as well. Now, if an animal or a person is infected, a sandfly will bite them, and then it can then transfer the protozoan from that individual to the next individual that it bites. Now, 90% of cases are reported from just a handful of countries, particularly Brazil, Ethiopia, India, Kenya, Somalia, Sudan, and South Sudan. There are estimated to be hundreds of thousands of cases per year, perhaps even up to 1 million, and tens of thousands of deaths. And there's two main forms of Leishmaniasis, mucocutaneous, which tends to cause ulcerations and lesions, particularly around the mouth or ears or nose, and the visceral form, which attacks the internal organs. And leishmaniasis, visceral leishmaniasis in particular, can have a potentially high fatality rate, whereas mucocutaneous leishmaniasis can cause disfigurement and can actually become visceral later. Now, when my boss first proposed this project, as I said, I wasn't super keen on it. It just wasn't really in my line of research at the time. But one day, all of that changed. We were on one of our field trips to catch bats for my main project. And just a warning here, I'm going to put up a photo that's a little bit graphic. So if you're kind of squeamish or sensitive, you might want to look away for a minute. I won't leave it up for very long. But when I was in the field, we caught a bat that looked like this. And even just from some of the background reading I had done on leishmaniasis, I could tell that it was very likely that this bat had leishmaniasis. This kind of ulceration on the ear is a typical location where those lesions would happen, and this doesn't look like the kind of wound that a bat would get from fighting with another bat or even being attacked by a predator. And we can actually see on this back ear as well, there's some scarring there. So I've changed the picture now to something that's not quite as graphic. So at the time I saw this bat and I thought this has got to be leishmaniasis, and that told me that this was probably a project that really was worth pursuing. Now, if I remember correctly, I actually released that first bat, but we went back to the same site and found another bat that also had similar lesions on the ear as well as on its leg, on the membrane next to its leg. And so I continued. We also received several other bats from the local animal control, and we tested them for leishmaniasis. We tested a total of 5 different bats, and we found that 2 of them were positive, and that included the individual with the lesions. That was a Pallas's long-tongued bat, as well as a velvety free-tailed bat, Molossus molossus, that didn't have any lesions or apparent symptoms. Now, we also found that the bat that was positive and had the lesions also had liver tissue that was positive for Leishmania. So that indicated that perhaps this had also, this infection had become visceral, which is something that we can also see in people. Now, since this study, which was published in 2013, there have been other students at the same university, the Federal University of Mato Grosso do Sul, that have continued these studies and identified more species of bats in more locations that also carry leishmaniasis or carry Leishmania. But we still don't necessarily have a very good idea of how important a role bats might play in the ecology of this disease. So there's still a lot more we have left to learn. So now I'm going to switch gears a little bit and talk about one of the projects from my PhD on modeling to better understand disease spillover from bats. Now, spillover is the transmission of a pathogen, could be a virus or a bacteria, from one species to another. And we use it a lot of times to talk about pathogens that pass from an animal, particularly a wild animal, to people. And our particular question was if bat diversity could explain where Ebola spills over to people. Now, I'll give a bit of background on where this question came from. So this was partially or largely inspired by the dilution effect. This was a rather elegant idea that was proposed in the early 2000s that explained how biodiversity itself could protect people from disease. And this was actually largely based on studies of Lyme disease in New England. And so the idea is that if we have an ecological community with lots of different species, and these species vary in their susceptibility to a pathogen. Some of them are more susceptible and some of them are less. But because you have all of these different individuals from the different species, the pathogen sort of gets spread out among them, and the overall prevalence, if we look at all of the animals present, is lower. But if we disturb this area and lose the biodiversity, and we might only be left with, and we're left with fewer species, which also might be more generalist and also more susceptible to the pathogen, then the overall prevalence in that area will increase. And there's also, here to mark the traditional perspective, I would say maybe just an alternative perspective, that on the other hand, biodiversity might actually amplify disease risk. And this is largely if we consider that all the different potential hosts are equally susceptible to a pathogen. If we have greater diversity, we have more potential hosts, and therefore the overall prevalence of the pathogen might increase. And so with this in mind, I was thinking about the relationship between Ebola and bats. And so this was something I had read about when I was working on the project on Leishmaniasis, and first sort of dipping my toes into the literature and research on bats and diseases. So Ebola virus was detected in fruit bats in 2005. And since then has been detected in 12 different species of bats in 3 different families. So that includes this fruit bat here on top, our leaf-nosed bats, and in free-tailed bats. Now, the potential distribution of all of these species from which Ebola has been detected is huge, ranging from the Middle East all the way into Southern Africa. And one thing I was particularly intrigued by was this bat, the Egyptian rosette, the Egyptian fruit bat, because it was a species that I had actually worked with, but in Israel. And so the idea that it was a potential host for Ebola in perhaps another part of its range was really interesting to me. And so if we look at the potential distribution of all of these host species, we can see that the spillovers are only occurring in a relatively small proportion of this potential range. I was also interested in understanding the role of bat diversity, because a lot of studies of modeling Ebola outbreaks tended to only consider a few species of fruit bat, even though Ebola had been detected at similar prevalences in those other species as well. And finally, there was talk in both the academic and popular articles about the role that deforestation might also play in outbreaks of Ebola, particularly on the 2013-14 outbreak that began in West Africa. And so with this in mind, I collected data for relevant variables I thought might explain where Ebola spillovers are occurring. And this included bat species richness, so that was all the potential bat species that could be present in an area, human population density, road density, crop cover, and pasture cover. And then we used 2 different types of ecological models to compare these 5 variables at locations where Ebola spillover has occurred to random background locations in sub-Saharan Africa where Ebola outbreaks have not occurred. And our models pointed to bat species richness as an explaining variable for where Ebola outbreaks occur. And our 2 models were quite consistent. So on these graphs, we can see along the x-axis, as bat species richness increases, the relative probability of an Ebola spillover event also increases. And this even makes sense if we just look at the map on the left. As the color becomes more green, the bat species richness increases, and we can see a lot of those X's where the outbreaks have occurred tend to be in areas that have more bat species. And Why might bat diversity influence where Ebola outbreaks occur? Well, studies have shown that zoonotic diseases tend to emerge in areas of high biodiversity. We've seen studies have also shown that the diversity of mammals appears to increase the risk of zoonotic disease spillover. Viruses that have greater host diversity are also more likely to spill over, and the incidence of viruses increases where many hosts coexist in the same area. And we also found one other variable that could help explain where Ebola outbreaks occur, and this was pasture cover. But here we actually found as pasture cover increased, the relative probability of Ebola spillover decreased. Now, once we knew that bat species richness was an important variable in understanding where these spillovers occur, we wanted to know if any particular family of bats might play a particularly important role. Again, the fruit bats have received sort of the most attention, but we've also detected Ebola in both free-tailed bats and leaf-nosed bats. And so we took the same approach where we compared the species richness of these different bat families in locations where Ebola spillover has occurred to locations where Ebola has not spilled over. And we also compared those models to our models of all bat species richness. And here we got a real surprise because we found that the diversity of slit-faced bats, Nycterid bats, could best explain where these Ebola outbreaks occur. And again, we see a really consistent response in our models, where as Nycterid species richness increases, the relative probability of spillover also increases. I said this was really surprising, and this is because Ebola has never been detected in Nycterid bats. But on the other hand, very few species have actually been tested or screened for the virus. One idea we had that could explain this pattern we see is that Nycterids are occasionally consumed. Now, some people have pointed to the consumption of bats, particularly fruit bats, as a potential driver or cause of Ebola spillovers, but other researchers have argued that consuming those types of bats is so common that if that was really the cause, then we would actually see many more outbreaks than what we do. But perhaps the consumption of a much more rarely consumed bat could maybe explain some of that discrepancy. Nycterid bats also can roost in buildings or in caves that people enter. So this is another way that people could potentially come in contact with a virus. Or these Nycterid bats could roost with other potential bat hosts or near other animals. For example, some species might roost in the burrows of aardvarks. And of course, because this was based on modeling, maybe we're actually picking up on something completely different, and Nycterid diversity is correlated with some other important factor or some other type of non-bat host. And there's actually been a couple of papers that were published recently. These are open access and free to access, discussing these ideas of potential other hosts other than bats of Ebola virus. Leave that there just for a second. And these results were published recently, just earlier this year, in the European Journal of Wildlife Research, and this is accessible on my website. Now, I've talked a bit about the potentials and pitfalls of a modeling approach, and now I'm going to go back to some field work and talk a bit about virus discovery in bats of Eswatini. This was another project within my PhD. Now, Eswatini is a small country in southern Africa, nestled between South Africa and Mozambique. And this study was carried out in the northeast part of the country. We trapped bats at 8 different sites, and we trapped those bats using mist nets. So these are really fine nets that we put up in places where we think bats might be flying, as well as harp traps, which is a slightly different trap with basically 3 lines of fishing line, and the bats try to navigate through them, and they usually can't get through, and then they end up falling in that bag below where we can get them. Once we had caught the bats, we placed them in cloth bags for a short amount of time until they pooped, and then we collected that poop and extracted DNA and RNA and sequenced it. Now, we managed to collect samples from 44 individual bats, and these belonged to 8 different species. We had 3 species of free-tailed bat, 1 species of tomb bat, 1 species of fruit bat, and 3 species of vesper bat. And we found a lot of different viruses in these bats. This included papillomavirus, picornavirus, parvovirus, circovirus, and herpes virus. This multicolored circle here, this donut, is one of the outputs from our analyses, from our bioinformatics, where we're comparing the sequences that we found in our samples to other known sequences. And everything that's in this sort of pink and salmon color is actually a virus. And as we get further out in the circle, the identification of those viruses becomes more specific. We also see a bit of green, that's some bacterial DNA that snuck through, and the blue bit is just a tiny bit of animal DNA, probably mostly from the bats themselves or any insects they might have eaten. But we also found coronaviruses. This was back in 2015 when all of us probably knew a lot less about coronaviruses than we do now. But I'll still give a little bit of background. So there are 4 main types or genera of coronaviruses: alpha, beta, gamma, and delta. Alpha and beta coronaviruses originate in bats. Gamma and delta coronaviruses originate in birds. And beta coronaviruses have included the viruses that cause SARS, MERS, and the current COVID-19. Alpha coronaviruses are known to cause some colds in humans. Now, bats are the sort of ancestral or ancestral host of coronaviruses. But these coronaviruses that we find in people are not the same. They might be related to viruses that we find in bats and share a common ancestor with the viruses we find in bats, but they do not, as far as we know, come directly from bats. And in some cases, they might also pass first somehow from bats to another animal and then infect humans. This was the case with MERS, which was spilled over to camels, and then people became infected through contact with those camels. Now, we found coronaviruses in half of our sites, in 4 of the 8 sites where we caught bats. They're highlighted on this map in red. And we found there was variation in the prevalence of coronaviruses in our different families of bats. So overall, we had about 18% prevalence. We found no coronavirus in tomb bats. We detected coronaviruses in 16% of our free-tailed bats, about 22% of our fruit bats, and 26% of our vesper bats. But again, we have to be a little bit careful because these are quite small sample sizes. And so a lot of the coronaviruses we detected were short bits or fragments, but we did actually recover some full genomes. So the full genome of a coronavirus is about 25,000 to 32,000 base pairs, compared to millions for animals, for example. Now, 3 of those full genomes were found in the bat Caraphon pumilus, the little free-tailed bat here. And what was interesting about these coronaviruses, they were all quite similar to each other, but not exactly the same. And we actually found that the two coronaviruses that were detected in bats that we caught at the same roost on the same day were more similar to each other, over 99%, compared to a third genome that we detected in a bat caught about 1 month later, about 10 miles away. And they were still very similar, about 98.5% similarity. And these are probably a strain or belong to the same species that has already been detected in the same genus, Caraphon, but not the same species in these Caraphon bats in Kenya. And so this might be a coronavirus that's widespread in these types of bats in southern Africa or sub-Saharan Africa. In addition, we found one other full coronavirus genome from Afronycteris nana, the banana bat. And this coronavirus was more different from other known coronaviruses. Its closest match was a coronavirus detected from bats in Italy that belonged to a different genus. Okay, so all of this talk of coronaviruses brings me to COVID-19. Now, I'm not an expert in coronaviruses. I've done some work with them, and I'm also not an expert on COVID-19, but I'll just talk a little bit about what we know so far. So it's important to note that COVID-19, SARS coronavirus 2, is a virus that infects people. It's a disease of people. It's likely that the ancestor of SARS coronavirus 2, the actual virus that causes the disease, COVID-19, came from rhinolophid bats. Right now we have a bat, a virus that has about 96% similarity to SARS coronavirus 2. But we have to remember that we also share 96% similarity in our genome, for example, to chimpanzees. So we're not talking about the same virus. And we don't know for sure right now what that transmission pathway was, how the virus came to infect people. It was likely one single interaction of a person with, right now, an unknown animal that had this virus. And the SARS coronavirus 2 likely diverged from this ancestor found in bats about 40 to 70 years ago. So that's a lot of time for a lot of things to happen. So we don't find SARS coronavirus 2 in bats, and we also don't find the same virus we find in bats in humans. So they just shared an ancestor, probably 40 to 70 years ago. But there's a lot of research still being done, and in our understanding and story of SARS coronavirus 2 is changing all the time as we learn more. Now, we do know that people can actually transmit SARS coronavirus 2 to animals. There's the case of the tiger at the Bronx Zoo, domestic dogs, domestic cats, although this is my cat and she thankfully does not have COVID-19, as well as mink in Europe and North America. And this is probably because SARS coronavirus 2 uses an ACE2 receptor to infect cells. And so the ACE2 receptor of these species is one that the coronavirus can use, and is somewhat similar to that in humans, although I'm definitely not an expert on receptors. And bats also have an ACE2 receptor that might make them susceptible to SARS coronavirus 2. And so I've been working with the IUCN, or as a part of the IUCN Bat Specialist Group. We have a special working group to address this issue. And with our, we've come to believe there's a credible risk of bats being able to be infected by SARS coronavirus 2. And in light of that, we've come up with a set of recommendations and guidelines to try to protect bats from exposure and infection to this virus. And we started our recommendations for researchers. They're mostly based on minimizing contact and handling of bats whenever possible, assessing whether or not you might have been exposed to SARS coronavirus 2, and if so, really avoiding any contact with bats, and if contact is absolutely necessary, adopting practices such as using a face mask, gloves, and frequent disinfection in order to protect bats. And we've also expanded our recommendations and guidelines to other stakeholders, including bat rehabilitators. I know that was something of interest to one of our participants here tonight. Cavers who might encounter bats, as well as guano collectors. And these infographics are all in English, but we've also translated them into a number of other languages. And we've also come up with a decision tree tool, particularly for researchers to help make decisions and avoid exposing bats to SARS coronavirus 2 in order to protect them. And all of these infographics, as well as the guidelines, are freely available on the website of the Bat Specialist Group. Okay. So now I'm going to switch gears a little bit and talk about my current work on the ecology of antibiotic resistance and show how we can go from studying bats and their ecology to understanding antibiotic resistance in hospitals. So antibiotic resistance is a huge problem. It causes thousands of deaths around the world every year. It's a huge problem globally, including here in Europe, where I'm currently working. And there are two main drivers. If we simplify a lot, we can say that there's two main drivers of antibiotic resistance. On the one hand, we have selection pressure. So that's mainly exposure to antibiotics. If we have bacteria that are exposed to antibiotics, the susceptible bacteria will die, and any that happen to be resistant will persist and then multiply and then kind of take over. And on the other hand, we have transmission of resistant bacteria. And in a hospital network, we can think of this as transmission between patients, patients and healthcare workers, visitors and patients, or surfaces, common surfaces. And for each of these two drivers, we have two different strategies. One, to lower selection pressure, we use strategies of antibiotic stewardship, so that's reducing the use of antibiotics, perhaps cycling antibiotics, or reducing the amount of time that we use them. And for to control transmission, we use infection control, so that can be screening of patients as well as cleaning and disinfection. Now, we know that these two processes are both really important for driving antibiotic resistance, but we don't know the relative importance of each of them, and this can prevent us from coming up with the most effective strategies to combat antibiotic resistance. And at its core, antibiotic resistance can be thought of as an ecological problem. So we often think of it more as a health or medical problem, but ecology at its base is understanding the interactions between organisms and their environment. And so we can use the tools of ecology to better understand how resistance is influenced by the environment around them. And so we can adapt many of the same models that I talked about earlier, for example, modeling Ebola outbreaks to the question of antibiotic resistance. And another thing I wanted to note is a lot of times when we discuss the ecology of antibiotic resistance, we're talking about antibiotic resistance in the outside environment, for example, in the soil, in water, in agricultural runoff. But the hospital is also an environment. It's a man-made environment, but it might be arguably the or one of the most important environments for antibiotic resistance. We have a lot of selection pressure from a lot of consumption of antibiotics, as well as susceptible patients. And this is where most people who contract antibiotic-resistant infections contract them in a hospital or healthcare setting. And so to understand how the hospital environment might affect antibiotic resistance, we use the idea of a metapopulation, which is just a fancy word for a population made up of smaller populations. And here we can see a few variations on that theme. Now, I'll explain in a bit more detail. So this idea came out of a bit of a contradiction. If we look at a very fine scale, we can see all animals have habitat that they need. Here I've chosen to represent our organism with bats, and all of these bats live in these patches of forest habitat. Now, the population of bats in each individual patch might actually be very unstable. We can see a local extinction or colonization. And all of these different patches, they vary in their size and their quality. For example, if they have good food for the bats to eat or the shelter they might need. Now, even though each individual patch is quite unstable, because they're connected to each other through the dispersal of individuals from one patch to another, the overall population is stable and persists. And so we adapted this idea to the hospital environment, where our habitat patches were the hospital wards, the habitat patch size were the number of beds, the connectivity between our different wards where the patient transfers between them, and we considered antibiotic consumption and ward type as indicators of habitat quality. So if there's a lot of consumption of antibiotics, that means a good habitat for resistant bacteria because it's killing off the susceptible competition. And we applied this idea to data collected from 357 different hospital wards in 4 different hospitals here in Lyon, France. And we considered over 13,000 ward admissions, and we used diagnostic antibiograms to quantify the number of infections in each of these wards. And we use similar models to understand the influence of each of these variables. So here I'm showing our results on 2 different types of resistant infections. In light blue, infections that were resistant to 3rd-generation cephalosporins, and in purple, infections that were resistant to carbapenems. And we might say that carbapenem-resistant infections are more resistant than cephalosporin-resistant infections. So if an infection that's resistant to carbapenem, that's one of our last-line antibiotics, and so there might not be a lot of other options for treating that infection. And here, what I'm on this graph, and I'll explain each one in a bit more detail. If we look, for example, at antibiotic use, we can see for every doubling of antibiotic use, we see about 5% more infections of both cephalosporin-resistant infections and carbapenem-resistant infections. And so this means the more we consume antibiotics, the larger the number of infections we see. Connectivity, we see a slightly different pattern where the connectivity doesn't seem to have much effect at all on the number of 3rd-generation cephalosporin-resistant infections, but we see actually a very similar effect, almost this 5% increase for every doubling of connectivity for carbapenem-resistant infections. So this tells us that those infection control measures might be almost as important as antibiotic stewardship for reducing the number of carbapenem-resistant infections. And we didn't see much of an effect for ward size or ward type, although we see a tendency for 3rd-generation cephalosporin infections to happen in these progressive care rather than intensive care wards. And then we also looked at the consumption of individual classes of antibiotics on the number of infections. And so here we see, quite logically, the more we use cephalosporins, the more infections that are resistant to cephalosporins. And this makes sense because that's a direct selection pressure for cephalosporin resistance, whereas we don't see an effect of carbapenem use on the number of cephalosporin-resistant infections. Similarly, for carbapenem-resistant infections, we see a positive effect of carbapenem use. So if we use more carbapenems, that increases selection pressure for carbapenem-resistant infections, and we see more of them, whereas third-generation cephalosporin use doesn't affect the number of carbapenem-resistant infections. But because this is ecology, we, of course, had a surprise in our results. And we found that the consumption of piperacillin tazobactam actually had a greater effect on both cephalosporin-resistant infections and carbapenem-resistant infections than either cephalosporins or carbapenems. And this was striking in part because piperacillin tazobactam has actually been proposed as an alternative to carbapenems in order to reduce the use of carbapenems. But we can see that this might actually have some unexpected consequences. We think we might see this result because piperacillin tazobactam is a very generalist antibiotic, and so it might be killing all of the bacteria. It's also taken as a pill, so it will affect the bacteria that are in the digestive system, and we have a lot of different bacteria there. So it might be killing off a lot of the susceptible good bacteria that we have in our digestive system, while leaving some bacteria that might actually be naturally resistant to antibiotics. And those can then be, because they're not in equilibrium with all the other bacteria, that could cause the patient to then have an infection. We also believe that this, we also saw an accumulation of small positive effects on many different species of bacteria. So here we've actually pulled together all resistant bacteria from 7 different species, and we include those bacteria that might be naturally resistant, not only those that have acquired a resistance. And because many studies tend to look at individual species, this effect on an individual species might be small enough to miss, but when we pool together all the possible bacteria that could have these resistances, we see this larger sort of cumulative effect. All right, so I tried to come up with something very profound for my concluding remarks, but I didn't quite succeed. So I'll just leave you with these few pretty basic thoughts. One, bats are the best. Ever since I first saw them up close, I've been fascinated, and I hope I've shown you how interesting and cool they are. I've also tried to show how ecology can help us understand these complex relationships and processes, and also that these tools from ecology can then help us understand the world around us, from bat populations to human diseases. So with that, I have this really long list of people to thank. So this is work that spans basically 15 years from when I was still in high school all the way through my undergrad, my PhD, and now my postdoctoral work. And none of this would be possible without all of these people who've also contributed to this work, as well as the different institutions where I carried out the work and the different institutions that funded it. And with that, I don't think I've gone too much over, so I think that leaves us time for questions. Thank you so much.
Thank you, Julie. That was really informative. My goodness. So if people have questions, if you want, you can use the chat window, or where it's such a small group, we might be able to just have people take themselves off mute and ask their question directly. So why don't we start that way? We'll just have people take themselves off mute and they can ask their questions that way. We have any volunteers want to start?
Yes. So I took myself off mute. Thank you, Julie. That was quite interesting. Two things struck me, and that was this requirement that to handle bats, you get the rabies vaccine, because obviously nobody survives rabies. And the fact that we're trying very hard not to include bats in a vector for spreading COVID-19. And just sort of wanted to get your thoughts or opinions, you know, it kind of reinforces some of that scariness, I guess, about bats, that you should be quite careful because of the transmission. And I guess, you know, if you compare it to other wild or even feral animals, it would be the same really with them as well. Or is there something special about bats? Perhaps they have more likely to spread disease than other wild animals?
Yeah, so thank you so much for that. There, in the last part of your question, you've definitely touched on sort of a big debate that's still going on sort of in the scientific and academic communities on whether or not there's something particular about bats that leads to having many pathogens. I wouldn't necessarily say diseases, which is when we're actually sick from something. There's been some recent studies that say perhaps the number of diseases that we see that spill over from bats is more a function of the great diversity of bats, and that if we sort of normalize between different types of animals, we could see that maybe there isn't, it's not remarkable, it's just that there's so many species of bats. Of course, all organisms, I mean, we have so many different viruses as well as just one species of human. There are other factors that could play a role, for example, the way that the different interactions people might have with bats, but I think it's still a bit too early to say whether or not it's just a function of the diversity of bats or if there's something particularly special. There's also been some studies on their immune system is a bit different because of their adaptations for flight. And therefore, for example, some of these pathogens such as Ebola don't appear to make bats sick, whereas it can make not only humans, but other animals such as primates and ungulates like gazelles sick. So there's still a lot of, I mean, I hate to cop out like this, but we still, I think, have more questions than answers. As far as rabies, I do want to address that one specifically. So basically all mammals can carry rabies, and the pre-exposure vaccines is really just, it's a precaution. And so people that work with other species of mammals will also have pre-exposure rabies vaccines. Veterinarians usually have pre-exposure rabies vaccines. And this is because if you're bitten by a bat, it could have rabies. It's unlikely, but you wouldn't necessarily know, and it would require testing the bat for rabies, which also involves killing the bat and checking its brain tissue. So in order to avoid having to do that, and also the quick turnaround time you would need in order to get that post-exposure vaccine, we get these pre-exposure vaccines. And we'll usually get like 2 follow-ups afterwards if we have an incident where we're bit by a bat, as opposed to like the 5 that you get if you don't have a pre-exposure vaccine. But I think the estimates of prevalence of rabies kind of vary a lot, but it's not higher than in other animals. It's just that you might not know, and yeah, you don't want to mess with rabies. So it's definitely better to just get that vaccine.
And are you familiar with the North American bat species? I mean, if we're here in Brookline, we don't have what do we have? Small brown bats and large brown bats or something, and not much else?
Yeah, I should have looked it up. I'm not exactly sure how many species there are in Massachusetts, but there are several different species. The little brown bats and the big brown bats are the most common ones. I don't think, you know, in North America, there's a big problem with the white nose syndrome, which is a fungal disease that has been killing bats, especially in the eastern US. So I think we don't, yeah, other than rabies, there aren't really any concerns for diseases from bats in New England or really in the US.
May I add?
I was just going to say, there was that article that was saying that the woman in New Hampshire was quarantining the bats because she didn't want to pass the COVID on. And I guess that was similar to what you were showing with your protocols. Yeah.
Yeah. And that's just as a precaution. Again, I mean, we know that people have given COVID-19 to other animals like cats and dogs and tigers and minks. And so we just want to avoid that. There's still studies kind of ongoing to try to assess the actual risk and how likely it is that could happen, but it's always best to avoid it. It's much easier to prevent something from happening than try to clean up a mess afterwards.
I wonder if I could ask about your Ebola research.
Yes.
You mentioned two of your variables were crop cover and pasture cover. And I was interested in why you picked those two as variables.
Oh, thanks. That's a great question. So originally, our original analyses were a bit simpler, and so we only included the road density and population density, and we felt that road density was a really good measure of human disturbance to an area that actually isn't strongly correlated with population density, and it also represents sort of the connectivity of people across the landscape. But we got some comments from other scientists who had read our article and the draft of it, and had suggested that maybe road density wasn't the only variable, or was maybe a bit too limited to account for anthropogenic disturbance. And so we decided to look at other variables that might also quantify disturbance. And we thought crop cover, where the planting of crops, and clearing of pasture were two variables that weren't captured by our other variables. For example, urbanization was quite well captured by population density. And so these were two other types of disturbance, that human disturbance that wasn't correlated with the variables that we had already chosen.
Thank you.
Does that make sense?
It does. It does.
Okay.
Thank you.
Thank you. Can get a little bit into the weeds.
It was very, very good. Thank you.
Julie, somebody asked a question through the chat, and they said they were a little embarrassed to ask you directly, but had you ever been bitten by a bat in any of your research?
Yeah, that's a good question, and no need to be embarrassed by any questions. All questions are good questions. And the answer is yes, and it was mostly my fault. So actually, that was one thing I wanted to mention as well, with the earlier question on rabies. You know, a bat is a wild animal, and so just like any other wild animal, if we catch it and we hold it, that bat might get scared, it might get stressed, it probably thinks we want to eat it, so its instinct is usually to try to get away, although occasionally we do have some individuals that are like really calm and chill when we hold them. And so there have been a couple of cases where I've been bitten. We definitely try to avoid it, but sometimes when we're working with bats, it might be really late at night, and so there's been a couple moments where I've just gotten a bit tired and a bit clumsy. For example, trying to put the bat in the bag where it'll stay until we have time to measure it, and sometimes I haven't done such a good job on that and been able to turn around and get a little nip in there, or not being careful enough taking it out of the bag or out of the nets. To avoid that, we use gloves, although that can make it really hard to manipulate some bats. If they're small with gloves, you kind of lose some of your dexterity. And then, yeah, and then the gloves can also kind of get caught up in the nets. So we definitely do everything we can to try to avoid it, but sometimes it still happens. So I've definitely gotten a few bites. And I'll also say some of these bats are like really, really small, like 2, 3, or 5 grams. And sometimes even though they bite, they might not be able to actually go through the skin.
One of the things that I'm a little curious about, and I apologize, I don't know a whole lot about bats, so this probably is going to seem like one of those silly questions, but you know, why are people so afraid? Like, where did that come from? You know, why are people afraid of bats?
I should probably do a bit more research on sort of the history and anthropology of that. I think it's largely because we don't necessarily understand them. And also they're animals that are out and about at night. We can't get, you know, until we start trapping them, it's hard to get a good look at them. And so I think also sometimes when people, describe to me having seen a bat flying around, you know, sometimes they'll estimate the size of what they saw. And I'll just know from like the area that they're telling me this about that there's no way that the bat could be that big. So I think, you know, it's just that we don't maybe see them or understand them. There are these misconceptions, you know, that they might be dangerous or have rabies, or even when I was younger, that bats will get into your hair, which, I mean, they definitely don't do that. There's also, you know, it's interesting, the myths about Dracula and bats and drinking blood, which came from Europe where there are no vampire bats. I think those kinds of ideas maybe led to people's fears. There's other parts of the world, particularly in Asia, where bats are considered like a sign of good luck. So it's also not universal.
Oh, that's interesting. Let's see. In terms of education and career choices, this person is asking about your seeming pivot from Romance languages to ecology, and that Romance languages have been beneficial to your travel and studies. Could you talk a little bit about that?
Yeah, for sure. So to me, I mean, it always seems like a pivot, but to me, it wasn't really a pivot, but sort of maintaining interest in a lot of different topics. So I was always interested in kind of in animals and working outdoors, and so I had these 2 jobs at Mammoth Cave National Park and with the BLM. That was when I was still in high school, but then I got to college and there were so many interesting classes in like social sciences and the humanities that I was as interested in as biology. And sometimes, I don't even know if I should say this, but to me, sometimes those classes from a, you know, studying and reading perspective could be a bit more interesting to me. And so I ended up deciding to major in Romance languages. So I studied Spanish and Portuguese, which was hugely beneficial. I mean, I did a lot of my work I ended up doing work in Brazil, and that was largely because I had studied Portuguese, and it made the experience completely different than if I hadn't had that language competence and allowed me to meet people and work with people who I might not have otherwise worked with. But at the same time, I also continued getting experience in biology in the lab, and I actually had a minor in biology. So I worked in a lab with fungi all for like 3.5 years of my undergraduate. And so I had a little bit less classroom experience and a bit more practical experience. Although once I started my PhD, I definitely had a lot of catching up to do in other aspects, such as statistics and programming. So it definitely helped in a lot of ways. And I think it helped again to, yeah, to take the classes, but then to also get the experience on the side along with that. I think understanding, you know, politics and history and like the history of science is also really important, or kind of the context in which science is done, and who does the science and what science we're funding is also just as important as the actual science that we do day to day. So I think it's really relevant for scientists as well to have that background in social sciences and humanities.
That's excellent. Do we have any other questions?
Oh, I'm gonna stop the screen share now. I'll also put in the chat. I have a website, and all of my kind of the publications and projects I've done are accessible through that. So I will put that there, so you guys have that.
That's great. I can, I'll also share that with everybody, you know, following the lecture. I'll send an email out. But this has just been fascinating. This is one of those there, I feel like I'm going to be thinking about this topic for the next few hours, and then I'll have all these questions that will come up. But really fascinating work and impressive research that you've done, and it seems like it's going to inform, you know, a lot of things, that the antibiotic resistance and infection control, and there's just so much to glean from all of that. And you know, the information you shared about bats, I'm even more intrigued by them. I didn't know half of, you know, what you shared with us, so I really have learned a lot, and I would imagine that everybody else that's participating in this feels similarly. Thank you so much, Julie, for your time, especially with the time difference. It's very late in France right now. I would assume it's well past your bedtime.
Well, it's midnight, and you know, I work with bats, so I'm a bit of a night owl. So this isn't much later than I'm usually going to sleep. So,
well,
so happy I could join you all today.
Yeah, thank you. And again, I'm just so proud of you. So very proud of you. And you know, thank you for all of your good work, and you know, keep it up, and we'll all stay in touch. Maybe we'll have you come back again for another lecture at some point.
I would love to.
All right, everybody, have a great evening. Thank you for joining in.
Thank you, everyone.
Take care.